This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1257, recorded on September 26, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Howdy, Vincent. Here we have 92 degrees and sunny. So, the first day of fall is was a couple of days ago, right?
Yeah, it's done.
So, not yet. Though I have to say, it was 64 this morning, which for here now is really a relief. And we're looking at mid-60s in the morning for the foreseeable future, low 90s. I'll take that. That's okay. I can handle that.
Here it's 27 C and cloudy. No sun to be seen. Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it's 80 Fahrenheit, 27 C here. Little bit of a breeze and clear skies, which is a nice break from the pouring rain we had for a few days, although we definitely needed the rain. So, not too bad today.
Shout out to all the people I met on my travels who are fans of our work. First at the Harvard Virology Retreat, which was in Waterville Valley, New Hampshire. Very rural and bucolic. But the license place, they say, live free or die.
And as George Carlin pointed out, you got to be wary of a state that mentions death right on the license plate.
I heard a Carlin skit the other day where he was bemoaning the inability to curse on TV and radio. He said it's all because of one pastor years ago in Mississippi who got offended, or Missouri, whatever it is.
Yeah, I was actually I changed my pick because things sort of resolved, but I was actually going to re-pick Carlin's monologue that I picked some time ago, 7 words you can't say on television.
Anyway, also a shout out to all those European virologists at the European Society for Clinical Virology. Many, many, many people came up to me and thanked us for what we do. So we're grateful for that. Every year, more and more people are listening, and I think that's great. And if you think it's great, we'd love your support because we don't put advertising on these programs. That's the main way that a podcast would get supported. But I decided a long time ago that we should have listener donations. And so if you go to microbe.tv/contribute, you find a number of ways by which you may contribute to our work. And as you know, in these times, we need good science out there, and we are one of many programs that do that in many different fields. Microbe.tv/contribute. Now, we are a virus podcast here on TWiV. But you know, the business between autism and Acetaminophen is an example of how to interpret scientific results. And we're good at that, right?
We do an okay job.
I hope so.
And when we're not, we correct ourselves.
That's correct. Which is, that happens.
And acetaminophen, for those of you in other countries, that's paracetamol.
Paracetamol, yeah. And so, you know, the RFK Jr. Has announced that he suspects that acetaminophen taken during pregnancy leads to autism. Trump, in fact, just said, don't take it. The FDA is going to put a warning label on the drug, a possible association between autism and children and the use of the drug during pregnancy. But if you're interested in why this is happening, there's a very good article in Nature by Helen Pearson and Heidi Ledford. Trump links autism and Tylenol. Is there any truth to it? So they're all talking about Tylenol, which is a stupid thing to do in the first place because that's just a brand name for acetaminophen, right? So, first of all, probably they can't pronounce it, so that's why they say Tylenol. And stocks of J&J, who makes the drug, dropped, and then they went back up again. But this article is good because it summarizes most of what is going on, but not all of it, actually. There have been many studies who have looked at the link between the two. Acetaminophen is the only drug available for women during pregnancy for pain and fever. And so it's important to be able to take that. And so it's been studied, and there's really no evidence that it causes autism. And most of the studies may find a slight association, but as these individuals in this article point out, there's so many confounding factors. If you just take people who take or don't take Tylenol during pregnancy, there's so many other things that distinguish them that it's very hard to say it's the acetaminophen that's doing that. And so there have been many studies that are doing this. And here I just want to point out the most robust research does not link autism and acetaminophen. In pregnancy, the better controlled studies are less likely to find even a small risk, said this scientist at Boston University. And even then, we're talking about a minor association. We do not think taking acetaminophen is in any way contributing to autism.
So the better you do your statistics, the worse the link looks.
That's right.
Yeah,
Confounding factors are an even bigger problem, which I just told you. Yeah.
My understanding is that, this is like, similar to the vaccines and autism thing, that there's a very large number of studies. And the study that is singled out, that part of what motivates this announcement is one study, a smaller study, that probably finds a stronger association than other studies. But my understanding is that particular study is overwhelmed by the bulk of the other studies that can't find an association. And the lay public should be very familiar with this sort of thing, where there's a whole bunch of clinical trials, and some say, yeah, maybe, and some say, probably not. And under those circumstances, what you can say is that there's no convincing association, okay? And that's typical. When there's clinical trials for something like this, when there's no association, it's negative results, right? So people have trouble with that. But my understanding is that all the clinicians and all of the relevant societies are adamant that there's no association. Okay. Based on the studies.
It's certainly, we actually know there are genetic links to autism, and the science is solid on that, and this rises nowhere near that level. So
acetaminophen was first made in 1878, but wasn't widely used until the 1950s. And autism was first described in the 1930s.
Because before that, the kids who had it just got shunted off somewhere, or, the family just dealt with that, and that wasn't really... this is true of many mental illnesses, actually many illnesses in general, the diagnosis only came up in the 1950s or something. Yeah, because we were actually doing clinical science in the 1950s.
Yeah.
The other thing that's worth mentioning is that, I think we've mentioned this before, but I think it bears repeating, is that it's frequently cited that the incidence of autism has increased. All right? And the reason for that, all of the experts agree, is because the definition of autism changed some time ago so that you could classify people on a much broader spectrum of diseases as being on the autism spectrum. Okay, so it's a classification thing, a diagnosis thing, not an actual increase in the disease.
Yeah. Which is actually, it's good that we have diagnoses for these things now, and it means we're getting more information and potentially finding the right kinds of help for people instead of just saying, oh, send them off to an institution somewhere. They got something wrong with them. So that's progress. But unfortunately, it has this side effect where people who want to make some story about it will come up with something and say, oh, it's caused by the phlogiston, or it's, maybe it's the crest or whatever.
So there's another side issue in this that I believe is brought up in this Nature article, and I've seen it elsewhere, is that it has this announcement has the unfortunate effect of putting the onus on the pregnant women. And this harkens back to a period from the 60s where a person named, Bruno Bettelheim from the University of Chicago, was promoting the idea that autism was caused by basically bad mothering, okay? Refrigerator mothers, cold, distant mothers. And that really caught fire for a while, apparently. Ultimately disproven, but caused a lot of pain and anxiety in people. So I can't imagine what it would be like if you're confused about this. First of all, am I going to take Tylenol or not? The answer is yes, go ahead. But still, it's got to be niggling in the back of your head because of this. And then what happens if you take Tylenol and have an autistic child? Are you going to blame yourself for the rest of your life? I sure as hell hope not. But that's one of the dangers of this kind of thing.
If you don't take Tylenol and you suffer through fevers and pain during your pregnancy, and then that's not leaving you in great shape for raising a kid. And that's the wrong direction.
But I think even if you think there's a slight association in any of these studies, there are certainly many children born with autism whose mothers did not take acetaminophen during pregnancy. So there's obviously are different.
And many, many, many children born whose mothers did take acetaminophen during pregnancy and who the kids are not autistic. So there's just...
we probably ought to get an autism person on the show sometime. That would be interesting.
Good. Yeah, that would be good.
But, my understanding is that it's extraordinarily complex mixture of genetic and potentially environmental factors as well, and we don't know what it is. And not taking Tylenol during pregnancy is not going to have an effect.
No.
And I would ask the listeners to ask themselves why this is happening. I try not to speculate on the air on motives. But I think it's worth asking why this is happening.
Why are we suddenly...
Why is the president of the United States making a medical proclamation, in particular, when it is based on a misinterpretation of the science?
Yeah, that's the key. The science does not agree with what he said.
Yeah.
And, this is dangerous territory for politics because science has the fortunate, or for some unfortunate, character of telling the truth. Okay, and you can't get around the truth. It's gonna be true no matter how much you don't want it to be true. And even if you're dead when it happens, it's going to be proven true. So it's not nice to mess with Mother Nature.
Yeah.
On
to the science.
That was all science.
I will just point out that it's worth bearing in mind that anytime, some announcement like this is leading the headlines, that's a time that other things are not leading the headlines, like Epstein or the Middle East or something like that.
Or misuse of the Justice Department.
Yes.
All right. Yes, this is all science, and now we're going to talk about some papers,
Yes.
which is how science is promulgated. And we have two today. The first is our snippet. This is an article in Nature Communications, this is a few years old, and I'll explain, creating resistance to avian influenza infection through genome editing of the ANP32 gene family. Alan, would you do the honors with the authors, please?
Yeah, so the first author is Alewo Idoko Ako. The co-senior authors are Wendy Barclay and Mike McGrew. And these folks are from University of Edinburgh, Imperial College London, Royal Veterinary College London, Pirbright Institute, UK, and the National Institute for Biological Standards and Control in Potters Bar, UK. So it is an all-British team.
So obviously this is relevant to the current circulation of avian influenza viruses, right? But it came up this past week. I was in Philadelphia, which, Alan, you're right. It's a wonderful city.
It's such a great town. Yeah.
I like it a lot. It's easy. It's calmer. It's not as busy. I really like it.
Yeah.
We had a textbook retreat to finish up the main. We're basically done with the writing of the 6th edition of the virology textbook, Principles of Virology, and this came up. And I looked at it and said, wow, this is really interesting. Even though it's October 2023 published, really interesting story, and it's relevant to today. You know, just because a study is old doesn't mean it's bad.
And 2023 is not that old.
2023 is not all that old.
Yes, you are right. It's not all that old.
I realize that time moves pretty lickety-split nowadays when we're living in the not even 24-hour news cycle. I find myself checking the New York Times on my phone, at 1 hour intervals, thinking things are going to change, right?
Time doesn't change all that much. You have to check other things to get it.
I want to point out a couple of things off the top with this paper. First of all, this and the other paper we're talking about today are both open access. And second, I love that the first sentence suggests that maybe they listened to TWiV, because not a lot of papers do this at the top. Influenza A viruses are enveloped, negative sense, single-stranded RNA viruses. I thought, yeah, this is great. We're teaching good habits.
So as you know, avian influenza viruses are a big threat to poultry, right? I mean, they're a threat to people and mammals now, but chickens are infected and you have to cull them, millions and millions of chickens. And so, they mention here that we can vaccinate, but it's not reliable because the viruses change, right? Antigenic drift. And they say it's controversial due to political and economic implications.
Yeah, so, I kind of stumbled over that. Economic.
I get.
I get. Political, I don't quite get. I don't even want to think about it.
So there are two things I can think of. One is just the general anti-vaccine tide that we have.
I don't want to eat a chicken that's been vaccinated.
Right. The other is international trade. It may be, and this comes up a lot in agriculture, both animal and plant, that you vaccinate your chickens so we won't know whether they might be carriers, and therefore we can block imports and have a non-tariff trade barrier that protects our farmers. So that kind of thing happens a lot.
Okay.
So they're looking for other ways, and this was a very interesting one. This has to do with the viral enzyme, the RNA-dependent RNA polymerase, which copies the viral RNAs, okay, because the cell can't do that. And that RNA polymerase, it's composed of 3 proteins, but it also requires a host cell protein. They're called the ANP32 family of proteins. It's the letter A, the letter N, and the letter P. It's not the grocery store ANP.
No.
It's ANP. ANP32A and 32B. And there's a difference between these proteins in mammalian and avian cells. The avian protein has an extra sequence, and it's needed apparently for the avian influenza virus polymerases to work, because when an avian influenza virus infects a mammalian cell, the ANP32 proteins don't work unless the virus changes to adapt to it, but we'll get to that later. All right, so a avian influenza virus going into humans, the RNA polymerase doesn't work efficiently because the AMP32 proteins don't work well. All right.
I cannot resist a nerdy sidetrack.
Sure.
You may not even be old enough for this, Vincent.
Hmm.
So the notion that a virus co-ops a host cell protein to do something that host cell protein may not even be designed for ordinarily to carry out its function, when it first surfaced, was really a striking phenomenon. And it still boggles my mind that these things evolved to use host proteins that maybe for some entirely different purpose to do their own thing. I believe, if I'm not, well, let me ask it this way. Do you remember, Vincent, when that first showed up? I think you may have been in high school. Or we're not that much different. We're a little different in age.
I was in high school in the
This would have been early 70s.
early 70s. Yeah.
Like 72, 73. Q beta replicase.
Q beta replicase. It's got a subunit from
It uses
right?
two transfer factors from protein synthesis,
Yeah,
TU and TS. Okay, along with a replicase in order to do its job.
Remember that, sure.
And I don't know it's probably been worked out what they do. I'll bet you they have RNA binding activity that's relevant or something like that. But I remember when that came out. I think that's probably the first example of this, and it blew my mind.
So
Sorry, had to do
the influenza virus polymerase requires ANP32 proteins, and as I said, the avian influenza viruses won't work in human cells. They also don't bind to the right receptors, but that's a different story. But if they did bind the right receptors and get in, the polymerase wouldn't work well. However, as you know, influenza viruses vary greatly, and mutations can be selected that will allow the avian polymerase to work in a human cell. And the first one that was discovered was described by Andy Mealy, who was on TWiV when we were in Wisconsin not too long ago. I think you were even there, Alan, for that one, right?
Oh, the ASV in Wisconsin, yeah.
I think you flew out there, in fact, maybe.
No, I went on the airlines for that one.
Okay. And also reassortment can provide the right polymerase sequence from another virus and so forth. So it's a problem. And but in this paper, we're looking at the chickens. So the chickens have ANP32 proteins that work really well with avian influenza viruses, obviously. And by the way, if you're wondering what ANP32 proteins do, they help make a dimer of the polymerase protein, so two heterotrimeric polymerases. So the polymerase is made of 3 proteins, and 2 of those, so 6 proteins in all, come together, and ANP32 helps to do that, to help form this asymmetric dimer. And there are amino acids that are known to be critical for this interaction. And the chicken AMP32B protein does not interact with the human viral polymerase. Right? It does with the bird, but not the human. So that's the background for what they try in this paper. They want to know if you introduce these changes into the chicken ANP32. All right. So people have looked at ANP32 and looked at what amino acids are critical, and they found these 2 changes that will block interaction of the avian influenza virus polymerase with ANP32. So they say, what if we introduce this into a chicken, a live chicken? Would it block influenza virus reproduction? And this is all made possible because you can easily edit chicken genomes and make chickens out of them.
So we're going to make CRISPR chicken.
CRISPR. Yeah, that sounds good.
That's delicious.
Sounds really good. So they use CRISPR-Cas9, this repurposed defense protein for editing, to introduce these 2 amino acid changes into the ANP32 of chickens. So they make a 2 amino acid substitution, and they make some cell lines with these. So remember, these 2 amino acid substitutions have been shown before to prevent avian influenza viruses from reproducing in avian cells. So they make avian cell lines with these 2 changes. And then they look at viral RNA polymerase activity in these edited chicken cells. All right. And they also have a line that doesn't make any AMP32A protein. So this is AMP32A. Chickens have 3, A, B, and I think E is the other one. But we're starting with A. And if you've been thinking and listening, you may say, why just A? Yeah, that would be a good thing to think about. But we start with A, and they made the 2 amino acid changes. Then they also made one which doesn't make any of the protein at all. Okay, so then they take, and then the human adapted RNA polymerase has a change at amino acid 627 of the PB2 protein to a lysine, and that makes avian influenza viruses grow well in human cells. Right, use the AMP32 of humans. Okay, so all these. They test avian influenza virus polymerases in the wild-type cells. They all work really well. But when you introduce these 2 amino acid changes at positions 129 and 30 into chicken AMP32A, no polymerase activity. Right? So we're just looking at polymerase activity on its own by not using virus. It's called mini genomes, which you can put into cells and just measure polymerase activity. Okay, so then they wanted to make chickens, okay? So they can do this. I'm not going to go through the technology, but they can take cells and they can modify genetically altered cells and introduce them into chickens, and they get chickens born who look okay. They have no developmental abnormalities. They don't have any differences in growth, external appearance, behavior, vaccination response, and they lay eggs.
They actually have a picture of modified and unmodified chickens in figure.
I love it.
1D.
They look the same.
I
right?
was looking through. I got to the data. I was like, oh, and they show the chickens. Great.
It's cute. It's very cute. And
Yes.
the only difference is a band around their leg, which, of course, the humans put on.
Right.
I wonder if you can take chickens to the opera.
Yeah, right.
And how they would feel about it.
Only if they have to cross the road.
All right, so then they're going to infect these genetically engineered chickens with avian influenza virus. So they have 10 wild-type chickens. They take 10 of these AMP32A modified chickens. They introduce virus intranasally, 1,000 PFU, and they're using a low pathogenicity avian influenza virus, right? Just because you don't need to kill all the chickens. You just need to know if the virus is reproducing and spreading among them. And they put some 10 chickens, which are called sentinels, in the cages to see if the virus will transmit from the inoculated chickens, right? They collect swabs, oropharyngeal swabs, and they measure virus by plaque assay. And all the inoculated wild-type birds, the virus reproduces, they can shed it, and the co-housed sentinels all get infected, right? It's transmitted from the wild-type birds. The AMP32 mutated chickens, they don't find any shedding in 9 out of 10 birds. One started delaying shedding from 4 to 6 days, and the titers were very low, and none of the co-housed birds got infected. Oh, it looks pretty good, right? It looks like this change really knocks down avian influenza virus infection. They serologically confirm that the birds have been infected. They can measure antibodies. The wild-type birds, they all make antibody responses. And these mutated birds, the AMP32A mutated birds, they don't seroconvert, except for this one bird that suddenly made virus later on. It didn't have very high titers.
Now, what kind of selective pressure is this putting on the virus?
Always pressure, right?
Mm-hmm.
It's always a lot of pressure. So they said, let's try a higher dose of influenza virus. Let's do 1,000 times the dose that we used in the first experiment.
Do you suppose that's reviewer 3?
Oh, I think probably thought of this.
I
They
think they thought
this
of this
themselves. Okay.
I mean, the first thing you ask when you've got something resistant to something is, what do the escape mutants look like?
Well, not only that, but there is that one bird in the first experiment
Yes.
that showed
that
a
was
little bit of
suspicious.
activity. So
I mean, I
that
would
would
have
make you
sequenced-
suspicious.
I would have sequenced that. Maybe they did, and then I don't know, but they don't mention that. So they give them a million PFU, and of course, all the wild type birds get infected, and they transmit to the sentinel birds. So that's not surprising.
1 million PFU is a hell of a dose. I
Yeah.
wonder what a natural dose is. Probably
Not a
nothing
million.
close to that.
I think it may be 10,000 or 50,000.
No, actually, I used to have a slide that showed how much human influenza there was in a microliter of sneeze. I forget what it was, but it was a pretty high number.
So as for the AMP32A mutant birds, they got low-level shedding at this higher dose in 5 of the 10 birds. Not all of them, so it's still a nice barrier. But they get low-level shedding, and the titers are pretty low, over 2 logs, 100-fold lower than what we found in the wild-type birds, except a single bird who had a titer of 1,000 PFU per mil. Aha!
Mm.
Wonder what that is. And this resulted in transmission to a single sentinel bird. So this shows that there is an impact on replication and transmission in chickens. So they say the ANP32A changes, the 2 amino acid changes, suppresses viral infection and limits onward transmission in naive contact birds. All right, so then they want to know whether any changes have occurred during viral infection. 2 amino acid changes is nothing. Easy to overcome. Now, they're not looking in the birds, of course. They're looking in the virus for changes that overcome those changes. So they take 6 directly inoculated birds, given this high titer inoculum, and they sequence the viruses that they shed. And they find that in all the birds who got infected at this high dose, the ANP32A mutant animals, they have amino acid changes in the polymerase proteins, sometimes alone, sometimes in combination. So they introduce these changes again into the mini genome, which you can put in cells, and it will replicate, and you can measure RNA polymerase synthesis. And so they find that the ANP32A mutant cells can support RNA polymerase activity with a virus that just has 1 amino acid change in the PA subunit. It's E349K that they picked up in this screen. And they make different combinations of these amino acid changes. That's the best one, it turns out. And they say that this is over. So there's a change in the virus that is overcoming the change in the AMP32A proteins. Right. So they make a virus that has 2 amino acid changes, and they co-infect eggs with this and with wild type virus, and they find that it maintains a reasonable frequency, which they say shows that those changes don't debilitate the fitness of the virus.
Right. So they did a competition assay where they infected with both wild type virus and the one with the changes, and the mutated virus seems to maintain its proportion of the population.
Right. They also did it in human airway epithelial cultures, and these double mutant viruses become enriched to 50%, so that it has a little replicative advantage over the wild type virus. So where are these amino acid changes occurring? So there's some published structures of the polymerase together with chicken ANP32A, and they can see where the polymerase is interfacing with ANP32A, and so they can they can see the PB627, which is in the domain that interacts with AMP32A, and so that's how it allows interaction of the avian polymerase with the human AMP32A. But this substitution E349K, which they picked up in their mutant selection, which has the biggest effect on polymerase activity, it's not located in any region that interacts with AMP32A. But they find that there's also an alternative dimer complex. And in fact, they find that these amino acids that they've identified in this study are located on that dimer interface, so between the AMP32A protein and the polymerase. So presumably that's how these amino acid changes are allowing good polymerase activity even with a mutated AMP32A or AMP32A in avian cells with an altered sequence by making changes that restore that interface. So then they said, well, okay, we put 2 amino acid changes in, the virus can still change to use it. What if we took out AMP32A entirely? Which you can do. You can delete it very easily. So they do that. They delete the AMP32A gene, and they make chickens with lacking AMP32A, and they seem to be fine, right? They look normal. They grow normal. They lay eggs, and then they inoculate their AMP32. They inoculate AMP32A 2 amino acid change eggs with 100 PFU of virus. They get a low level of viral replication, and they get polymerase mutations, amino acid changes in those. So again, confirming that these 2 amino acid changes don't completely abrogate virus reproduction. And of course, in wild type eggs, everything is normal. But in the knockout eggs, the double mutant still replicates. The virus with 2 changes that they selected in a previous experiment here, it still replicates in I meant to say mice, animals that lack AMP32A.
So how's it doing it?
Deletion of the entire AMP32A is not enough to block avian influenza virus infections of chickens.
Of chickens.
Why am I saying mice and children instead of chickens? Okay, so
You're just now used to talking about chickens, Vincent.
No.
I'm not. I don't talk about chickens very much. So there are other AMP32 proteins in chickens, right? There's AMP32B and AMP32E. So they make chicken cells.
And we don't know what happened to C and D.
Yeah, we don't know why they skipped C and D. They make cell lines where they have lost production of AMP32A and B, AMP32A and E, or all 3. Why not? When you're making one, you might as well make all 3. So TKO is the total knockout.
Now we're talking about cell lines, not chickens.
Yes. Not yet.
Okay,
These are cell lines.
The presumption is that the chickens will not be well with double or triple knockouts.
So again, you have avian influenza virus with changes in the polymerase that allow it to use mutated AMP32A. So you can find replication of that virus in chicken cells that lack AMP32A. We already knew that. And if you have only 32E present, they will still replicate. But when you take away all 3 AMP32 proteins, now that virus does not reproduce.
We finally got it.
Finally got it, yeah. And so they then they use virus replication assays, because what I've told you so far is a mini replicon assay, again, which is just measuring polymerase activity. But they want to know, in the context of a virus infection, do you get the same result? And they do. So you got viruses with these amino acid changes that allow it to grow in cells lacking AMP32A. You put them in the triple knockout, AMP32A, B, and E. Now they do not grow. So single edits aren't good enough. You need to have triple deletions. And they're cell lines, right? They're not chickens. This is all being done in cell lines. And they say that, I don't know who did this experiment, but they say that the chickens are probably not going to be very healthy with the triple knockout. That this combination of knockouts is expected to be deleterious to the animal's health. All right. But it illustrates a proof of principle that multiple edits could be combined to block virus reproduction.
Could be they tried and didn't get any chickens.
Yeah.
Maybe. It's too bad, right? But you can imagine that maybe they could introduce more subtle changes in each gene
Yeah.
combined, and that would do it, right?
The
Instead
next
of
thing,
deleting
the next thing to do is to find out how the viral polymerase is using ANP, B, and E. How's it binding, and can you make a couple of amino acid changes to muck that up?
Yeah.
That's
so you
another
could-
paper.
It may well have been done, in fact, in the last 2 years, but I just thought this was an interesting proof of concept. So you're going to make genetically engineered chickens that are resistant to influenza A virus infection, okay? So the first question is, can you do it? And we don't know until we do the experiments, but it's going to be done. People are going to do this experiment. The second question is, will people eat them?
Sure, I would.
I would too, and so would Condit, but
There are a lot of people who might not. And
who might not, yeah.
It also gets into the whole genetically engineered organism discussion, and there are whole segments of the population that think that that's bad. And yeah. So and also, there's going to be considerable cost in the research and development to get these chickens made and to raise large quantities of them and sell the eggs. Somebody's going to need to turn a profit from that. So if these flu-resistant chickens cost more than the normal chickens, then you're back to the economic problem, at which point maybe you could just vaccinate.
Well, they say that's not useful in many situations,
Right.
so I don't know what to do. That is a problem, because if you get in the U.S., if your flock is infected with avian influenza virus, you have to cull them all.
You're flocked.
Right.
Yeah.
So it's a problem. It's a big economic problem. So this is quite interesting that you can make chickens that would be resistant.
So I want to make sure I got this right. You guys can check me on this. But one of the things that caught my attention is that if I read it correctly, one of the main escape mutants. So avian influenza doesn't replicate in human cells for a variety of reasons, one of which is apparently that it doesn't interact with the human equivalent of this ANP32 protein.
Right.
However, if I read it right, the double mutant, the escape mutant from the chickens now will replicate or at least interact with, I don't know if it interacts with, but it no longer cares about the human ANP32. Okay.
Yeah.
So that raises it's not quite a red flag. It might be an orange flag to me.
Sure.
My first reaction to that is, so if we got these chickens that are engineered, would that evolve a virus that could now be better capable of replicating in humans? Because another issue about this whole thing is, one of the reasons we're interested in the avian flu is that its potential as a pandemic virus, if it could jump to the humans. So that we want to keep an eye out for, I think.
Of course, it's not a problem with the triple knockout chickens, but
No, it's
we don't
not a
have
problem with the
chickens.
chicken. But we don't have triple knockout chickens.
No, we don't. Triple knockout chicken cells. That's right.
No.
Right.
So then,
But,
that's a problem.
That in a way, that's neither here nor there. It's a good thing that came up and, could, think about it. But it's the proof of concept here that's interesting. Okay.
Hey, could we replace all the animals on the planet with animals that are resistant to all viruses?
That's going to be a big project.
Yeah, I think
Yeah, that's going to be a big project.
well,
There's
it's not possible, but you could go through every species. Of course, we don't know a lot of the
Well,
viruses.
they're the ones we haven't discovered, yeah.
But if, you
Actually-
could do it with avian influenza viruses first because they infect many different species
Yes.
now, right? It's
you know,
not just
actually,
chickens.
if that were possible, nature would have done it by now. I
Yes.
think nature,
yes.
nature's the best experimentalist. Okay, and
And has a 3 billion year head start on us.
had a 3 billion year head start, and the chickens are still getting infected.
The viruses are
Viruses are clever.
very clever. If we can describe cleverness, they are strongly driven by selection to exist, right?
Well,
and so
DNA and RNA really want to replicate, and
Yeah, it's all about
I don't
DNA and
fully
RNA.
understand the motivation there. Okay, but I take heart in the fact that the motivation is strong, and even if humans wipe out themselves and 90% of the other species, DNA is going to continue to replicate.
it's very interesting because in the RNA world, right, where there was no cellular life and just RNA, those molecules self-replicated.
Yeah.
Mhm.
So starting from there was a strong push to reproduce, and that evolves continuously and changes.
You don't have to invoke a motivation. I mean, the reproduction of it means that more will reproduce because that's what
Yeah,
reproduction is, right? And then you get more
Yeah.
copies of it, and that's how the whole process works.
Well,
really like
the motivation is thermodynamics, right?
Yeah.
Gives free
Thermodynamics.
energy.
Well, it's
Yeah.
just a chemical reaction,
Yeah,
basically.
that's right.
Right? It's all about chemistry, and
Right.
that's pushing it forward. Yeah, that's a good way to look at it. That explains life. That's it.
Delta G, man.
Yep.
Delta G. All right, now we have a paper, a cell paper, combination antiretroviral therapy and MCL1 inhibition mitigate HTLV1 infection in vivo. Alan, can you do the honors of the authors?
Sure. First author is James Cooney, and the co-senior authors, 4 of them are Lloyd Einsiedel, Damian Purcell, Marcel Derflinger, and Mark Pellegrini. And this group is from the Walter and Eliza Hall Institute of Medical Research in Parkville, Australia, University of Melbourne, Alice Springs Hospital in Alice Springs, University of Queensland, UCB Pharma in Smyrna, Georgia, and the Centenary Institute Medical Research Foundation in Camperdown, Sydney, Australia. So with the exception of the Georgian, this is an Aussie crew.
And the reason I picked this, it's an interesting paper, but also many people have written and asked to talk about HTLV, and we really have never talked about this retrovirus. So we've talked a lot about HIV, the other human retrovirus, but HTLV, human T lymphotropic virus 1, is also a human retrovirus. And, as retroviruses do, they have RNA genomes that are copied to DNA by a retroviral reverse transcriptase, and the DNA then integrates into the cell nuclear DNA.
This is the Rodney Dangerfield of human retroviruses.
Yes.
Don't get
Doesn't
no
get it-
respect.
Don't get no respect.
And so it's a kind of a cousin to HIV, and epidemiologically it's similar. It's spread blood contact, sexually, breast milk, so the
Yep.
kind of the same mechanisms. And there are, what did they say, 10 million people estimated in the world have this, so it's
Yep.
not a
Yep.
trivial problem.
5 to 10% of those at risk of serious
Right.
complications.
So most people everybody gets lifelong infection. Once you get it and it integrates, that's it. But only 1 in 20 people get disease. And one of those diseases is adult T-cell leukemia lymphoma, or ATL, right? Or neurological conditions, or lung problems, as you will see here. There are four types: HTL1, 2, 3, and 4. Only one apparently causes disease. They all infect you for your life, but apparently only one causes disease.
And some populations have a lot of this, and
Yes.
other populations don't. And so globally, it's not equally distributed. And a major reason, I gather that this group is almost all from Australia, is that one of the big populations of people who have this are Australian Aboriginal communities in Central Australia. For whatever reason, there's a lot of HTLV1 there.
Yes, there's a lot of variability in who's infected. It's only 1% of people in North America are infected with HTLV1, but in other countries, a third of the population or more are infected, including Australia.
That's got to be heavily multifactorial, but one of the things that comes to mind is you wonder about restriction factors in different populations or whatever.
And I mean, when you're infected, you don't get symptoms for many years. Decades after infection, right? And there's a very nice article in the Cleveland Clinic that we will put in the show notes, which gives you a rundown of all the diseases.
Cleveland Clinic and Mayo Clinic are awesome with these kinds of things. They're kind of my go-to resources for medical information. They're very good.
And one of the main targets of infection is CD4 cells, just like HIV-1, but with different outcomes.
HIV-1 kills them. HTLV-1 does not.
Yes. One of my favorite names for a disease is HTLV-1 associated myelopathy, or HAM, which is also called tropical spastic paraparesis, TSP. So the disease is HAM/TSP. 30 to 60 years after you're infected. Risk factors include injecting drugs or being born in an area where it's common. Like the highest rates are in southwestern Japan, sub-Saharan Africa, parts of South America, the Caribbean, northern Iran, and central Australia. I mean, northern Iran. I mean, why?
Why?
Right.
One of the things that occurred to me with this distribution is it may, as Rich said, there may be specific host factors, restriction factors that are common in particular genetic populations. Or it may just be that these are populations that happen to have somebody come in with the virus and they are otherwise fairly isolated.
Yeah.
And it just spread there and didn't really get out much. But that's a theory.
Now, the problem here is that there's not much to do about infection. They say preventive or therapeutic drugs are lacking for this virus. It's way behind HIV-1 in terms of we have many, many antiviral drugs used in combinations, right? And so,
my guess is that part of the reason for that is that it doesn't cause enough disease in enough people.
It could be.
So it doesn't get enough attention.
And also there are subtypes, right? There's HTLV-1 A or C, and C is the subtype found most commonly in the Australian Aboriginal communities. And they say this is strongly associated with chronic pulmonary disease, which is not found elsewhere. So they wanted to. First, there's no animal model to test therapeutics, so they wanted to do that, and then they wanted to use it to test some drugs. There have been use of some HIV antiretrovirals to try and treat HTLV-1, but they don't really work very well. And remember, you can treat people ahead of time, pre-exposure prophylaxis, or you can do post-exposure prophylaxis, and basically there isn't a good therapy at the moment, right? So that's what they're trying to do in this paper. So they have a humanized mouse model of which they make for HTLV-1 infected. So they start with humanized non-obese diabetic severe combined immunodeficiency IL-1 deficient mice. So these are extremely immunosuppressed mice that have been given human immune tissues. And then they take and they're used in many other studies. They weren't made just for this.
So it's basically a way of having a human immune system in an animal.
Right.
Right? But the animal's got to be severely immune compromised, otherwise it's not going to tolerate
That's right.
maintaining the human tissue. It's going to
That's
reject
right. That's
it.
why they use these mice. So then they take peripheral blood mononuclear cells from an HTLV1C infected donor. They irradiate them, and then they introduce them intraperitoneally into these mice. They're called HuNSG mice. Okay, so that what happens is they see a big increase in proviral load. So remember, the provirus is the DNA form of the virus integrated into the genome. So after giving these mice these infected peripheral blood mononuclear cells, they see a big increase in proviral load. The CD4 T cells expand. So all suggesting that these human mice are now getting an established infection. They get
So I had a question about the technique here, because they take the PBMCs from HTLV1 infected donors and irradiate them.
Yes.
So I figure the idea is you don't want those cells to
Right.
replicate. You want
Yes.
to kill those cells, but they're a source of virus, right?
That's right.
Okay.
That's right. Right.
So the irradiation is going to damage the DNA, but not the viral RNA.
Right. Okay.
So they can study who's infected with these and so forth, but then they take more irradiated HTLV1C positive CD4 T cells as an infectious inoculum to put into these mice and show that they get de novo infection in certain hematopoietic precursor cells. And that establishes chronic infection. All right. And so now they have a mouse that's chronically infected with HTLV1C. They can look to see where the reservoirs are. Blood, spleen, lymph nodes are major proviral reservoirs. They see lots of memory CD4 T cells. They see lots of regulatory T cells in these animals. And so they say we think this is going to be an animal model for studying HTLV1 pathogenesis and also antiviral therapy. The provirus in these, interestingly, in these animals, in these mice, the provirus, only 22% of the proviruses have intact viral sequences. So you get lots of deletions in these animals. But most of them retain the TAX gene, which is thought to be, it's a transactivator that's thought to turn on oncogenes in the cell, and that leads to the tumors. And so this is consistent with what happens in people. They retain the TAX gene, but delete lots of other gene as well. They also see, they find that the proviral expansion and the lymphocytosis is higher than what you see in humans. And I think, well, that's because the mice have a crappy immune response. And they try other mice backgrounds for doing this with less severe immunosuppression, but that doesn't help. But that when they add antibodies to the virus, to the mice, then it restricts the lymphocytosis and the viral expansion. So it's showing that this is happening just because it's an immunocompromised mouse.
Do I understand this correctly, that one of these pieces of data here say that in these infected humanized mice, virtually every CD4 positive cell is infected?
Yeah, I think that's correct.
It's I would call that a robust model.
Yes.
Yes. Yes, it's a really good model. Yes. Yes, it's almost all of them. Not all of them, but many, like 67% have one piece of viral DNA.
Oh, I saw 67% with one, 29% with two.
With two.
With
Right.
two.
3% with three, and then there's a couple of 0.7s here pointing to something, and I'm guessing one of those is the zero, or there isn't a zero. I don't see any T cells here.
Effectively, everything's infected.
Effectively,
Yeah,
everything's
it's true.
infected.
It's true. All right, so they say we have a mouse model that recapitulates aspects of human disease. We have cellular infection, clonality, proviral gene expressions similar to what's in humans. Disease is characterized by features of ATL the lymphoma rapidly progressive lymphocytosis and organ infiltration, including the lung. So they're going to use this now to look for intervention strategies for HTLV1. So the first thing they do is to try and understand if there are any subtype-specific differences, and they're going to compare HTLV1C and HTLV1A, which are the most divergent subtypes. And 1C, again, is the prevalent in the Aboriginal Australians, where you get that unusual lung disease. So they infect mice with both of these subtypes, and they find that they see a slightly more aggressive disease phenotype in the 1C infected animals. They also look at proteome changes. They see infection associated with high levels of many cytokines, right, reflective of inflammation. And they say these are responsible for inflammatory diseases, very characteristic of the ATL. And that's what they also see in the HTLV1C and HTLV1A infected animals. And this lung disease in humans is associated with a high proviral load, infiltration of T cells into the lungs. So they harvest and stain organs from HTLV1A and HTLV1C infected animals. And they find that both subtypes can drive inflammatory lung disease, but the 1C infected animals seem to have a more aggressive disease. So they see differences in infiltration of cells in the lung, in the liver, and the kidney, for example. So some indication that the 1C in the mouse model is slightly more aggressive, as it is in humans. They do single-cell RNA sequencing to try and understand how HTLV1 modulates the transcriptional landscape of the host, and basically they say that there's a lot of concordance between the transcriptional response in their mice, in the T cells, compared with HTLV infected ATL patients. So they say this further says that this model is good for reflecting what's happening in people. All right, then they go on to antiretrovirals here. And they say no attempt has been made to reduce HTLV1C transmission in these indigenous communities in Australia.
Which I don't know if they meant that literally, but it seems to me like a colossal failure of public health if nobody's gone, like at least an educated people about risks for transmission, and maybe you ought to mitigate that. But if not, then that's a problem. Or if they just mean that there aren't any good drugs for it, then okay.
So they note that sexual intercourse is the major mode of transmission in this community, 40% for females, 70% for males. And then mother-to-child transmission by breastfeeding, a further 3% contribution to transmission. So they wanted to use their model to look at antiretroviral compounds, which have all been developed for HIV, to look at early infection, right? Because they would like to be able to give it to people and prevent infection. So there's an antiretroviral called tenofovir, which is a nucleoside analog. I believe, is it a nucleoside analog?
I think so, yeah.
Yeah, it inhibits the reverse transcriptase. So it will inhibit the HTLV reverse transcriptase. So it's an HIV-1 approved drug. And if they give it as pre-exposure prophylaxis, this tenofovir reduces infection of HTLV1C compared with controls, using doses that are equivalent to the human doses. Right? So that's good. So 58.3% of mice were free of infection compared to 17% for the controls. The control they use is another reverse transcriptase inhibitor called 3TC, which is used in combination therapy for HIV-1, but which doesn't work for HTLV-1. It doesn't protect mice against infection in their model, so they actually use it as a negative control, which is cool. All right, so that's preventing infection, right? It's pre-exposure prophylaxis. So then if you have an established infection, what will this drug do? So that's post-exposure prophylaxis. So they ask whether it can inhibit when given after infection. So they take their mice, they take PBMCs from who? HTLV-1C infected PBMCs, I guess from humans. They put it into their mouse model by intravenous injection. They establish an infection. Then they give them tenofovir. And they say tenofovir marginally impacted the de novo spread compared with the control. So they said maybe we need to use a combination and target different parts of the reproduction cycle of the virus. So then they use an integrase inhibitor, dolutegravir. Integrase is the viral enzyme that takes the double-stranded DNA made by reverse transcriptase and integrates it into the human DNA, or the mouse DNA in this case. And if they use combination tenofovir and dolutegravir, it reduces significantly expansion after challenge, right? So 6-fold lower proviral load across the time. So monotherapy isn't good for therapy, but 2 drugs seems to work a little better. All right, so then it's known that infection with HTLV-1 messes with apoptosis, programmed cell death. So programmed cell death is a response of cells to stress, including virus infection, which kills the cell. And the idea is that you kill the cell and prevent virus reproduction, so you save the organism. You kill some cells and save the organism. It's thought that HTLV-1 somehow interferes with programmed cell death, so the infected cells survive, so more virus can be made, right? Makes a lot of sense. So there are 2 proteins that are involved in the regulation of apoptosis that are targets. So one of these is BCL2, which promotes apoptosis, and this protein is downregulated in HTLV-infected CD4 positive T cells. And then there's another protein, MCL1, which is an inhibitor of apoptosis, okay? And in HTLV-infected cells, MCL1 production goes up. So the stimulator of apoptosis goes down, and the inhibitor in the stimulator, the inhibitor goes up.
Right.
Pretty clever.
So that's consistent with the virus adjusting levels of these 2 regulatory proteins to prevent apoptosis.
Right.
The cell is trying to commit suicide, and the virus
won't
doesn't
let it.
want that to happen.
So they said, why don't we target these 2 proteins, these 2 cell proteins, BCL1 and MCL1. I have trouble with these acronyms. BCL1 I've thought about for years. That's fine. But MCL1 is a new one for me. So could they turn on apoptosis and kill HTLV-1 infected cells? Okay. So they have an inhibitor of MCL1. So remember, MCL1, I already forgot what it does.
MCL1 inhibits
anti-apoptotic.
apoptosis. BCL2 stimulates it. Yeah.
So they're going to inhibit the inhibitor of apoptosis.
Right.
And so that's MCL1. So they're going to give their infected mice an inhibitor of MCL1. And what they find is that the HTLV-1 infected cells are more likely to die. And BCL inhibitors don't work very well at all for whatever reason. They say, well, maybe these cells are only dependent on
BCL stimulators.
Stimulators of BCLs don't work. So MCL1 seems to be the thing that works, so that's great. So maybe this could be used to manage HTLV-1 infected animals. So they do that. This has been done in cells so far, what I've told you. So now they look in animals. So they treat HTLV-1C infected mice with this inhibitor of MCL1. And after a couple of weeks of treatment, the CD4 T cell pool, the infected CD4 T cell pool shrunk 3.5-fold after having expanded after you infect the mice. So there's a lot of drug-induced cell death. And if you don't give them drug, if you give them a control, the HTLV-1C infected T cells continue to expand. Now, interestingly, they gave the same drug to uninfected animals. It has no effect on CD4 T cell numbers, right? So that makes sense because it's the virus that is driving the inhibition of apoptosis.
Right.
So if you don't have virus there, then there's nothing to mess with.
And this drug that they're giving, this MCL1 inhibitor, is just called S63845, which leads me to believe this is an experimental compound from somewhere, whereas tenofovir is an approved drug that lots of people take.
All right. So these infected CD4 T cells are more susceptible to the induction of death upon MCL1 inhibition. So then they ask whether MCL1 inhibition could prevent or mitigate proviral load expansion in vivo. So they give HTLV-1C infected PBMCs to uninfected mice. And then they're immediately given combination antiviral with tenofovir and dolutegravir, and then they're given S63845, the MCL1 inhibitor. After 3 weeks, 81% of the mice have no detectable proviruses. This is really good.
Yeah.
It is a mouse, but it's something that you would think about going forward with. So that's really, so you need combination. Now remember, you're going to infect these mice and then give them drugs quickly afterwards, 2 antiretrovirals and 1 targeting apoptosis protein, MCL1. But looks pretty good. So I don't know.
So that's the kind of intervention you might use for people who have a known exposure or highly suspected exposure, like healthcare workers who have a needle stick. It's not something where somebody comes into the clinic and they're already developing symptoms because 30 years ago they contracted this virus. You're not going to be able to use this.
So they have the possible intervention here.
Yeah.
They say these results support the initiation of trials to investigate the efficacy of these drugs like targeting MCL1, because that's what's not been tested. As Alan said, the antiretrovirals have all been tested and licensed in people. Well, there you go. That's the story. I thought would be interesting to let you hear about HTLV-1 and some attempts to prevent it. And it's interesting that this comes from Australia, where they have a huge problem, right? So that's what drives investigation. There's very little interest in the U.S. There are people who work on HTLV-1, but very few infections in the U.S. They're not zero. There are people, and they write periodically to us. Tell me about HTLV-1. What's going on? So here it is.
There you go.
This could be used for everyone.
So I don't know. I'm been digging around to try and find out about S63845.
Mm-hmm.
And I don't know who makes it, but it's under investigation as an anti-cancer drug. Okay?
Right.
Okay.
Avoidance of apoptosis is critical for the development of sustained growth of tumors. The pro-survival protein, MCL1, is overexpressed in many cancers.
Yeah.
Okay? So you want to target that dude so that they'll die.
Okay. Let's do a few emails. Alan, can you take the first one?
Yes. Volker writes, Dear Vincent and the crew, a couple of remarks on episode 1243. There was quite some Racanielling about RFK lying. How could he? I believe this is bullshit in the sense of Harry G. Frankfurt. And that's the book on bullshit, by the way, which everybody should read.
Which my son-in-law gave me early in our relationship. Okay?
So Volker continues, this bullshit in this sense. RFK simply doesn't care whether what he says is true or not, as long as it serves his purpose. In this case, I assume part of that purpose is to flood your, Vincent's, systems while dismantling the larger system. It took only 2 months to overthrow democracy in Germany in 1933. I watch in horror and disbelief what is happening in the U.S. today.
Volker,
In the
by
same
the way,
episode-
is writing from Germany.
Yes. In the same episode, all of you discussed the economic effects of people in science losing their jobs. Unfortunately, the arguments given, jobs being lost, would also apply if science provided no more benefit to society than digging holes in the ground and filling them up again. I am a bit dubious about economic claims like every dollar in basic research leads to X dollars in return, simply because economics is not a hard science, but rather a belief system, or even two distinct and competing belief systems. Did you mention a return factor of 11 recently? That seems much higher than what I found in published studies. What seems more solid is to compare government-funded research relative to GDP. To my surprise, the US is not spending much more than the EU. Both are around 0.65%, provides a reference. Therefore, significant cuts would seem detrimental in terms of international competition. In total R&D, public and private, relative to GDP, however, the US is far ahead of the EU, about 3% versus 2%, same source. Assuming privately funded research is indeed profitable, cutting the supply of scientists to the private sector and dismantling public trust in science might be the even worse effect of what is happening right now. Finally, thanks for reading my question on polio and sanitation, and thanks to the, quote, person who shall not be named, end quote, for the great answer. Excellent insights, including the correction of a former TWiV fact, no gut immunity by IPV, and even a mystery. Why does this expert not like to be named? I like it. Greetings from Bavaria, Volker. Yes, I assume the expert in question has a job that they would prefer not be linked to the comment.
Right. Exactly right. Yeah.
So
So
it's not Voldemort.
not
No.
Voldemort. No. Not Voldemort. Yes, the return on investment is a little flimsy, right?
Yeah.
I mean, I've heard 11 and I've heard 3. So, but as he said, the GDP, the funding related to GDP is important.
I like the idea that economics is a belief
Yes.
system.
Right. The economists won't agree with that.
Well, no.
Though I must say, my son-in-law, the same one who gave me the book on bullshit, okay,
Yeah.
is an economist. All right. And anytime I ask him an economic question, the first words out of his mouth are, "It depends."
Yes.
Yeah, it's say a lot of things about virology. Depends.
Yep.
Like, will this virus infect? It depends on the host. It depends
Yeah.
on the immune system. That depends on a lot of things. Rich, can you take the next one?
Anne writes, "Dear TWiV scientists, I very much appreciated your podcast, episode 1243, discussing a possible reason for my metastatic breast cancer. My original breast cancer was in 1991. It was stage 2 and treated with some rather harsh chemotherapy. It came back to my I'm sorry to hear this story. It came back to my lungs in October 2024, 33 years after the original diagnosis. I believe every woman who has had invasive breast cancer knows that it could come back. But after 10 years, I had stopped worrying about it, and certainly after 30 years, I wasn't even thinking about it, despite my unexplained shortness of breath. So now you know I found your report on viruses stirring up breast cancer cells in lungs very interesting. When I was diagnosed last October, a friend of mine studied the science of metastatic breast cancer and learned that science does not know where those cancer cells lurk, nor what causes them to reactivate. I had not had COVID, and it's been many years since I've had the flu or even a cold. But I have, of course, had flu vaccines and the mRNA COVID vaccines. I now have to wonder if those flu vaccinations woke up my cancer cells. On another note, in the early 90s, women got angry about the lack of research into breast cancer, and together we went to Congress and demanded funding for research. Not surprisingly, the only place there was a fat in the budget was in the Department of the Army, and they began a breast cancer research program that continues to this day. That research, which I'm hoping is beyond the reach of RFK Jr. since it's not in the NIH, led to the discovery of CDK4/6 inhibitors as treatment for metastatic breast cancer when combined with aromatase inhibitors for those of us who are postmenopausal and who have estrogen-positive cancers. The results are amazing. In my own case, 9 months of treatment has reduced the size of tumors in my lungs, and no new ones have appeared. Yay. That's my comment, the yay. Thousands of women are living many years with these treatments. But as my oncologist told me, all cancers mutate, and there is no cure for MBC. Eventually, the treatments will stop working. Curiously, most metastatic breast cancer appears first in the bones rather than the lungs. Apparently, about 30% of us have it in our lungs. I hope somebody studies how a virus would lead to a bone metastasis. Thank you for giving me one possible explanation for my MBC. With a shout out to Alan Dubb because I live in Amherst, Massachusetts and do my treatment at Bay State in Springfield. Ann. So, number one, I'll reiterate, Ann, I'm sorry to hear about your situation. Very much so. And I'm glad that if nothing else, we can be an outlet for you to talk about this. So write anytime you want. Now, didn't we have a letter that was from a researcher who gave an in-depth sort of reanalysis of the statistics in that paper and threw significant question into whether or not that there aren't these cells that hang around, but whether or not they are actually awakened by virus infection. So do I remember that correctly?
Yeah, and it was really about how significant a phenomenon is this.
Right.
Because when and as described in that letter, which was a couple of episodes ago, I think, they looked at the table and walked through it, and the numbers that you get to with people who actually had this, despite the enormous patient sample size, you end up looking at fairly small numbers of cases, and then you're extrapolating from those. So the data are maybe not crystal clear.
Yeah. So it may not have been your COVID. So
yeah,
from that letter made it clear that it's not definitive at all.
Yeah.
Right.
Right.
Yeah, and I certainly wouldn't blame, the flu vaccine or the COVID vaccine. Those seem like really unlikely triggers for this, just based on what we saw in the paper, even if this is a real phenomenon, which we now have to put an asterisk on.
Yeah. Charles writes, hello, Twivers. Hot, humid, and uncomfortable in Chapel Hill, North Carolina today. The link, which he includes, is to a nice opinion article that is better written than I could do about the recent Jay Bhattacharya Washington Post opinion article about why the NIH is turning away from mRNA research. Anyone putting faith in Dr. Bhattacharya's opinion should read his research. His paper about seroprevalence of SARS-CoV-2 in Santa Clara County is a telling article. Should be required reading in Epidemiology 101 classes. Great example of selection bias. By the way, Spotswood is in New Jersey near Old Bridge and New Brunswick. So this is a science-based medicine article by Jonathan Howard entitled 2,500 Commentators Agree Dr. Jay Bhattacharya has failed to earn the public's trust. And I did read this when the email came in weeks ago. It's really good. Let me see if I can sum it up. Yeah, he did this COVID seropositivity study during COVID. It was just totally...
It was ridiculous.
I remember looking at that.
Yeah.
I remember we discussed that at some point.
Yep.
So he wrote an article titled Why the NIH is Pivoting Away from mRNA Vaccines. It said, as a vaccine for broad public use, mRNA technology has failed to earn the public's trust, which I think is a lie.
Yeah.
For
To
God's
whatever
sakes.
extent it has failed to earn the public's trust, it is the fault of people besmirching it by making false claims.
Yeah. So then he goes through and he talks about, so he said, trust. What are the objective data for trust? How about efficacy in saving lives? Isn't that what vaccines are supposed to do?
Yeah.
This is what happens when we have an NIH director who has never cared for a patient in his life. He has appeared on Fox News, the primary criteria to have a seat in the Trump leadership circle. NIH scientists already regard Bhattacharya as incompetent. It's like a pyromaniac blaming the fire brigade for failing to have public trust in its capacity to prevent conflagration. He references no data for this. The problem with Bhattacharya's argument is that it mistakes public opinion for a legitimate scientific compass. Pure politics, zero science.
Yep.
Anyway, it goes on. It's really good. Lots and lots of bullet points. Yeah, I don't know how someone could write something that's just so wrong.
We're seeing a lot of that nowadays.
Yep.
I could never
We
do
just
that.
have to get through this.
Good Lord. Back to you, Alan.
Sure. Jay, I'm assuming not Jay Bhattacharya, writes, I see from the signature, definitely not. All right. Greetings to all you purveyors of fine science information. I've been a listener to your podcast since 2016, the year I also received gene therapy for hemophilia B. I had written to your show a year or so later to thank all your listeners, scientists, and you for all you do. At the time, I think I emphasized that your work matters. With the current state of science and the incredible amount of noise in between scientists and patients, I figured you might all need a morale booster. It's now been 9.5 years since my single 45-minute-long intravenous gene therapy treatment, and 9.5 years of carefree living, not even one bleed, as the community likes to call them. Yes, the work you do matters. I know that many people enter the medical science field with the crazy notion that they want to help people and contribute to a better society. Well, if you cut through all the noise, it's not a crazy notion. Your work mattered to me, it matters to me and others now, and it will matter to many more people in the future. Thank you all. Please keep going. Thank you, Jay, who's in Cambridge, Ontario, and that is great.
What a great story.
Yeah.
Now, of course, I'm burning with curiosity about the details of his treatment.
Yes.
Most of those are some sort of retroviral insertion of a good gene, right?
Yeah.
Ex
Right.
vivo,
Yep.
and then repopulate your immune system with that. Man, that's cool.
Very.
One-time infusion that uses a vector to deliver a functional viral gene. One time. Trying to see. So it's factor 9
Yeah.
impairment.
They
so
use
this, so it was approved in 2022. And then there are other in trials as well. It's not the only one that's going to be used. There are others.
Cool.
And the one that's been here is recruiting by Regeneron. Oh, there's no drug being tested. It's an informational study. Okay. Yeah, it's cool. I don't know what you think it's a lentivirus vector?
That's what I would suspect, yes.
Well, I'm going to look it
But I
up.
don't know.
I'm going to look it up while you read the next one, Rich.
Me?
Yeah,
David
you're
Your
writes.
turn.
David writes, guys, don't you get it yet? What we are witnessing is the revenge of the inept. The nerds had their chance decades ago. It's lunacy for sure, but these are the C- and D students getting us all back for our 4.0 excellence. I have had countless patients complain to me about the RMNA vaccines, one of whom came up to me shortly before his demise from COVID in 2021, telling me that he had just seen Joe Rogan kick Sanjay Gupta's ass. He died a few weeks later, his family praising my care. And he gives an obit to that. How we got here. The tyranny, hashtag the tyranny of merit. What's become of common good? And he gives a link to the book, The Death of Expertise, a campaign against established knowledge and why it matters, by Tom Nichols. Okay, have any of you read this book?
I have. It's a very good book. Yeah.
I've read Tyranny of Merit, which I'm actually just going to pull up a link.
Yeah,
A link to that.
The Death of Expertise is very good. Tom Nichols goes through all the reasons he thinks we are here.
It's worse than we think. And David is an MD in Syracuse. So, yeah, basically, if you want to promote your particular ideology or conspiracy theory, it helps to have a population that can't think critically.
All right. So for hemophilia B, I have my slide from my course. Yeah, it's an AAV vector.
Oh, that's cool.
Yeah.
It says it delivers clotting factor 7. So for A and B, there are 2 clotting factors that are defective. B is 9. One shot, $3 million.
Wow.
I presume the insurance paid for it.
AAV, isn't that interesting?
Yeah.
So, the retroviral vector technologies had an issue in that the integrations are random and can potentially set off bad side effects, cancers themselves. AAV doesn't integrate. I mean, this still boggles my mind. It's maintained basically as an episome indefinitely, right?
Yeah.
Okay. That's incredible. I wonder if they bleed this guy now and then.
Just to see,
Ask him to write back. Hey, where was he?
Ontario.
Which
means he
was
may not have had to pay $3 million for the treatment.
He may not have had to pay $3 million, but was it Jay? Yes.
Jay.
Jay.
Yes.
Jay. Do they follow up with you occasionally and look at the, status of your AAV vector in your blood cells? I want to know.
Yeah.
I want to know if it changes over time, how it's maintained, what your expression profile looks like. There's a lot of science there to do. How cool is that?
The trial that you were talking about, there was a trial for SCID, severe combined immunodeficiency, where the patients have a defect in the IL-2 receptor gamma chain. So they gave them a retrovirus with the wild type gene, and a couple of kids developed leukemias, T-cell leukemias, because of integration next to oncogenes, right?
Okay.
And those kids actually were treated with chemotherapy, and they recovered, so they're okay.
Okay.
But they since modified the vector so they don't integrate like that anymore, and they don't have a promoter, basically in the LTR that would drive it.
Well, interestingly, I think one. I'm sorry. Go ahead.
And they now do lentiviral gene therapy. So, for example, SCID is now treated with a lentivirus, and other diseases are treated with lentiviruses. But they use AAV in some cases as well. So you get the same effect. You get long-term correction because the lentiviruses integrate. The AAVs just persist episomally.
Well, and if I understand this correctly, the actually a motivation for investigating AAV to start with is that it also can integrate. But it turns out that that's only if you have the whole virus around, and that if you're just using it as a vector with the sequences that make it a vector, it doesn't integrate. But it turns out that's okay because it will persist extrachromosomally indefinitely, which still boggles my mind.
Why are doing a plasmid transformation on human cells?
This, right? I guess they just say, "Oh, DNA, nucleus, that's okay."
Okay, Gretchen writes, I listened to a recent podcast in which the question came up, is there a vaccine for cancer? And someone wrote in to say, yes, there is one for prostate cancer. I recently came across this article in Precision that presented findings from an initial clinical trial for 2 other types of cancers. I thought you might be interested to know. So this is in Precision Medicine. Vaccine improves survival in pancreatic and colorectal cancers. Mutated KRAS is a driver observed in nearly half of colorectal and the vast majority of pancreatic cancers. A novel vaccine targets the resistant KRAS gene in human tumors. Interesting.
Cool.
But it's just investigational.
Yeah.
By the way, you are my go-to weekly podcast. I learn so much from all your fine colleagues, and don't shy away from the political. It's important to call out every single lie. As a dual citizen, I'm concerned for not only our countries, but for the world with this assault on science. Ridiculous, dangerous, and downright criminal. Keep up the excellent work. Gretchen is from Toronto, so I guess you have a duel with the U.S.
Yeah.
As Alan says, though.
I would argue the hepatitis B vaccine and the HPV vaccine are both vaccines for cancer that are currently in wide use.
Absolutely.
Great
Matter of fact, I
success.
think those are the best examples.
Yeah. So Hep B causes liver cancer, or is a cause of liver cancer, and HPV, of course, causes cervical cancer.
Yeah, we have 2 more. Let's finish them off for Alan.
Sure. Mike writes, while in prison, he wrote about what was happening, which is available in the book Notes from Prison. Who are we talking about here? He described his theory of
Bonhoeffer?
stupidity.
No.
Notes from Prison.
Somebody
Okay.
named Bonhoeffer?
Alija Izetbegovic is the first hit that I'm getting. Bosnia and Herzegovina. Anyway, he described his theory of stupidity, which was outlined with these premises. Defenselessness against stupidity. Unlike evil, which can be confronted and resisted, stupidity is resistant to reason and evidence, making those who oppose it feel helpless. Stupidity and power. Stupidity can enable evil by allowing it to rise to positions of power. Powerful individuals often exploit the stupidity of the masses, using simple narratives and slogans to manipulate them. The role of power. Power itself can contribute to stupidity by encouraging conformity and suppressing independent thought. Individuals in positions of authority may become automatons, reciting slogans and losing their capacity for critical thinking. The need for critical thinking. Bonhoeffer. Okay, so this is Bonhoeffer, I guess?
Yeah, I looked him up. It's Dietrich Bonhoeffer, Letters and Papers from Prison.
Okay. Bonhoeffer emphasized the importance of courage, responsibility, and independent thinking to combat stupidity. He believed that fostering critical thinking and encouraging people to question information were crucial steps in breaking free from the cycle of stupidity. And finally, stupidity as a weapon. Bonhoeffer also suggested that stupidity can be a weapon used by the powerful to control the masses, making people willing instruments of a larger machine. Keep pushing back against the stupid with your educational efforts. Those in power are deliberately gathering support from those among us that believe stupid and dangerous ideas. You provide truth and information which will triumph eventually because reality can only be denied for so long. The only question is how long stupidity will be given power and free rein to destroy so much of what we took decades to put together. When this storm of stupidity is past, we'll have some hard work rebuilding what we once had. Thank you, Mike.
I think that's a nice summary of the episode.
Indeed.
Very, very good. That looks like a good read. So wait a minute. Why was Bonhoeffer in prison? Do we know?
I don't know.
That's another investigation.
For speaking out against stupidity.
Probably.
And you're the last one, Rich.
Okay. Robert writes, I really enjoyed the podcast about Frank Stahl. I met Stahl several times, and he helped me with different types of density gradients, including the cesium sulfate gradient for RNA, which is too dense of cesium chloride. Yeah, RNA pellets in a cesium chloride gradient. Got to use cesium sulfate. For your info, Stahl's thesis advisor was Gus Dorman, who I met when I was a postdoc at University of Washington. Wild type T4 is called T4D after Gus.
Did you know
And
that,
I'm
Rich?
gonna do the whole thing here. This is from Robert Sclafani, Professor Emeritus of Biochemistry and Molecular Genetics at University of Colorado, and a Fellow of the American Academy of Microbiology. Is this part of his letter?
Yeah.
A tag, you can observe a lot just by watching. Yogi Berra. I have a couple of colleagues who spent their careers at the University of Oregon. One, Karen Sprague, who told me that every time she sees Frank, or every time she saw Frank, little cesium chloride gradient peaks would come up in her mind.
Yep.
Great stuff.
All right, let's do some picks of the week. And Rich, you are first.
Okay, so let me get back there. I'm trying to do a search on Bonhoeffer. I'll figure it out. But okay, so I have an emerging infectious disease article that is majorly nerdy, entitled Chagas disease, an endemic disease in the United States. This pick is motivated by a zinger, which I will give at the end. Okay. The paper itself simply says that it's a group of scientists sort of summarizing the status of Chagas disease and where it's thought to be endemic, and noting that in Texas in particular and other places in the southwest United States, where there's been Chagas disease around for a long time, it is officially not regarded as endemic. Okay? But then they go through all the data about how much Chagas disease there is, how much of the vector there is, how many infections there are, what the definition is of endemic, and say, wake up, dudes. It ought to be ruled endemic in the United States, and that's important because it influences the attitudes. Okay? So, classic EID article. The zinger is, if you'll recall, some episodes ago, I mentioned that my yoga teacher is in epidemiology with the state of Texas. And she is, in fact, the penultimate author on this paper. Bonnie Mays is my yoga teacher. One of my yoga teachers.
Nice.
Who I will see tomorrow morning.
Upward-facing trypanosome.
There you go.
I want to comment, Rich, that I don't know exactly when this happened, but I noticed it when you started doing your pick, which is about Chagas disease. There's Dixon very clearly in the background.
Oh, yeah. Hi, Dixon.
Nice, yeah.
Yeah, good.
Yeah.
Mm-hmm.
This was one of his interests.
Alan, what do you have for us?
So I have an article from Science by John Cohen, typical John Cohen, nice long article that really delves into an issue. This is the long, strange trip of an archive of some of the most important artifacts in the history of molecular biology. What a good example of this contains the original Photo 51 from Rosalind Franklin, and this was the X-ray diffraction image from which it was possible to determine the double helical structure of DNA. Many, many other lab notebooks, drafts, images, a whole bunch of material, tremendous amount of stuff. This whole archive is now finally available to the public and researchers at the Science History Institute in Philadelphia. They recently bought it for an undisclosed amount from Craig Venter. I hope the undisclosed amount was something like $1, because Craig Venter doesn't really need the money. But what's worth an entire article about this is how this archive came to be. It was assembled by an actual legitimate archivist, rare book dealer, and this guy, Al Seckel, who was kind of a con man. Well, not kind of. He made up a resume and went around collecting this stuff when people didn't really know its full value. And his intent was to collect these priceless artifacts and sell them for a profit. And it's just it's really bizarre the route that, that this archive then took, and the multiple lawsuits back and forth, and all this. So really worth a read. And next time I'm in Philly, I'll have to swing by the Science History Institute to check them out.
Here's the slide rule that Rosalind Franklin used, engraved with her initials.
Mm-hmm.
Yeah.
Francis Crick's lab notebook, Nobel lecture, and a letter to fellow Nobel laureate John Kendrew. Cool stuff.
Yeah.
I just walked past this museum this week when I was in Philly.
Okay.
That's cool. I didn't know this. Hopefully I'll get back. All right, my pick is an article in Wired by Ritsuko Kawai. I don't know how you say it, Kawai or Kawai. But the world's oceans are hurtling toward a breaking point. We know how important the oceans are, right?
Yep.
But we're pushing them to their limit. They're in trouble. Overfishing, pollution, rising temperatures, acidification, and of course, climate change. And this is a very nice article summarizing that. And basically a modeling study from Santa Barbara, UC Santa Barbara, that projects by 2050 the cumulative pressure on the oceans could increase 2.2 to 2.6 fold compared to today, especially at the equator, the poles, and the coastal area. And this is open access, so you should go read it because it's real. It's real stuff, and it's really bad. And we're messing things up here on Earth. I don't understand why money has to drive everything, because that's what it is.
It's because it's short-sighted,
Yes.
No concern for the long run. But this is the ocean's revenge. The oceans will kick humans off the planet, and then they will revive themselves, and all will be well.
How are they going to kick us off? We're going to have to go somewhere
We
else.
won't be
Is that
able to
the
survive when the oceans go belly up.
Oh yeah. If
They won't go belly up completely. There's still going to be DNA replicating, remember?
end? Yep.
Okay.
Yep, remember.
And we'll just turn it over. Okay.
Good boy. So, we in this country, we had some good environmental regulations in place, right, to minimize, automobile emissions and other sorts of impacts. But they're being all taken away now because the current administration doesn't care. It's more interested in making money for companies. And it's very short-sighted, as you say, because in a number of years, it's going to be disastrous. It's not fair to future generations. Yeah, I thought humanity would be more understanding and sensitive than that, but apparently not.
So I have to tell you that Dietrich Bonhoeffer has a wiki page.
Yep.
He was born in 1906 and died in 1945. He was a German Lutheran pastor, neo-Orthodox theologian, and anti-Nazi dissident who was a key founding member of the Confessing Church. His writings on Christianity's role in the secular world have become widely influential, blah, 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 blah. So he was courageously vocal during the Nazi reign, and was imprisoned in 1943 in Tegel prison for a year and a half, which I guess is where he wrote that stuff, and ultimately transferred to the Flossenburg concentration camp, and then ultimately, shortly before the end of the Nazi reign, on 9th of April 1945, was hanged after a phony trial.
All right, that'll do it for TWiV 1257. You can find show notes at microbe.tv/TWiV. If you have comments or questions or picks of the week, TWiV at microbe.tv. And if you enjoy these programs, TWiV and all the others, TWIP, TWIM, Immune, Infectious Disease Buzzcast, etc., We'd love your support. Microbe.tv/contribute. Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, Dixon and I had a wonderful time here.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv, and you can find Microbe TV on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV. Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.